Hello everyone, welcome to my channel all about zoology. So today's video is about regulatory enzyme. Before going to learn about regulatory enzyme, we have to know about the enzyme. Okay, so what is enzyme? We know enzymes are protein that act as a biological catalyst by accelerating chemical reaction. Okay, and they are bind to a substrate molecule. Okay, on their active site. This is about the enzyme. Now here the regulatory enzyme. Regulatory means something can be regulated or modulated. Regulatory enzyme means enzyme whose catalytic activity can be modulated are termed as regulatory enzyme. Okay. So here the catalytic activity is what? Catalytic activity is basically the ability of a catalyst, or you can say an enzyme, to increase the rate of a chemical reaction without being consumed in the process. Okay. Then enzymes catalytic activity can be modulated either negatively or positively through structural alteration. This line is important. That influence the enzyme substrate binding affinity or turnover number. Okay. So here as the image or picture shown, there are an enzyme and substrate binds, and also there is active site present on the enzyme and form enzyme substrate complex. After that they produce two products. Okay, enzyme and product. Now, how regulatory enzyme works? So, the regulatory enzymes exhibit increased or decreased catalytic activity, as I said, in response to a special signal. It is important. Why? Something or some signals are present to regulate the regulatory enzyme. Okay, so in response to a special signal. And those special signals are of five types. The first one is allosteric regulation. The second one is feedback inhibition. The third is covalent modification. Fourth one, compartmentalization. And the fifth, cofactor and coenzyme. Okay. Now, how catalytic activity of a regulatory enzyme can be modulated? There are two ways. The catalytic activity of regulatory enzyme can be modulated through reversible non-covalent binding or reversible covalent modification. So first one is reversible non-covalent binding. There are a modulator present. Okay. Regulation through non-covalent binding or non-covalent interaction of an enzyme with regulatory compound, it's termed as allosteric regulation. Now we will learn about allosteric regulation. But here, as I said, a modulator present, and that modulator is known allosteric modulator or allosteric effector. They are of two types. Okay. They are of two types. Activator and inhibitor. Activator are a positive modulator type and inhibitor are negative modulator type. So activator is what? Activator means that enhance enzyme's catalytic activity. Okay. And the negative modulator or inhibitor is what? They inhibit enzyme's catalytic activity. Okay. They function as a two type of modulator, activator and inhibitor. Okay. Now what is allosteric regulation? Allosteric. This word is a Greek word. Allo means other, steric means place or site. Okay. Allosteric enzymes are a class of regulatory enzyme which increase or decrease catalytic activity in response to spatial signals. Its function through reversible non-covalent binding of regulatory compound called allosteric modulator. Modulator are of reversible and non-covalent binding type. Okay. So here allosteric modulator present. Most allosteric modulator or allosteric enzymes are oligomeric in nature. Oligomeric means they consist multiple subunit. Okay. Important point. Allosteric enzymes are maybe homotropic and heterotropic. Homotropic means substrate and the modulator are of same type or identical. Okay. But in case of heterotropic, they are non-identical. That means an enzyme, the substrate binds to the target site and the modulator bind to other site. Okay. But in case of homotropic, substrate and modulator both are bind with the same site. Okay. So heterotropic means the substrate and modulator are not identical, as I said. They have two functionally different binding site. Catalytic site and allosteric site. Catalytic site binds with substrate and catalyzes the reaction. Binds with substrate and catalyzes the reaction. Those are catalytic site. And the allosteric or regulatory site means binds with an allosteric modulator, not on the substrate side. It may be other side they will bind. Okay. So here an example is given. It is a less active enzyme. Two types of site present on that enzyme. First one is catalytic site and the allosteric site. Okay, catalytic site binds with the substrate and allosteric site binds with the modulator. Here it is a less active enzyme. After adding a positive modulator, it becomes more active enzyme. It is in substrate binding site, it is in modulator binding site, and that modulator are of positive modulator type. Okay, so this one is an example of allosteric regulation. I hope you understood. If you like this video, please don't forget to subscribe my channel. Till then, keep learning. Take care and bye bye.